which means, if you remember back to transcription, here's a gene, here's +1, this is the transcription start site, here's -10 with the tartar box. A transcription factor binds at the promoter, and so it binds at the beginning of a particular gene to turn that gene on. It was discovered by David Page and Lowell Goodfellow many years ago now, and with their work on mice. A pedigree chart for the Y chromosome would look like this. It's pretty straightforward. If you see a pedigree chart where it's only males, then it could be a Y chromosome inheritance pattern. So here's a male with a Y chromosome and it has a particular trait. Here's the female with X. Obviously there's no trait because she doesn't have a Y chromosome. This Y chromosome is passed to the son, and one of the X chromosomes is passed from the mother. The Y chromosome has that trait, and so on down the line. Basically every male in that particular line is going to have that trait if it's on the Y chromosome. If we now look at a few problems if things go wrong when we are passing on those chromosomes to the next generation. There's something called non-disjunction. Non-disjunction here means failure to separate in anaphase, which means that, let's say you have two X chromosomes and in anaphase 2 they fail to separate. This means that one gamete will have two X chromosomes and the other gamete will have none of the X chromosomes. Which means when you pass on this gamete, when it combines with, say, another gamete containing a single X chromosome, instead of just being 1 and 1, you have 2 and 1. Therefore, here are 3 X chromosomes in this particular female. So here's non-disjunction in a female egg cell, and we can see here we end up with a triple X female. If that 2X egg combines with the Y carrying sperm cell, then we have an XXY, which is a male, which has a syndrome called Klinefelter syndrome. And if we have the opposite, where we have the egg that's devoid of an X chromosome combined with an X carrying sperm, we end up with a female with just one X chromosome. This is called Turner syndrome. A fetus or an embryo with just a single Y chromosome and no X chromosome is non-viable, so this will die. This does not even start to develop. Here's a cartoon and photograph of somebody with Turner syndrome, and you can see there are a variety of different traits, multiple changes throughout the body, which would make sense because you are now haploid for all the genes on the X chromosome instead of being diploid for the genes on the X chromosome. With the Klinefelter syndrome, which is XXY, if you look at the karyotype here on the right hand side, you'll see here is the issue, and the issue could either be it's a male with an extra X chromosome, or you could equally say it's a female with an extra Y chromosome. And a lot of the phenotypes are similar in that they have both male and female phenotypes being present. A common trait due to non-disjunction is Down syndrome, which you have probably heard of. This is trisomy 21, which means there are 3 chromosome 21s. So a cell will have 3 copies of chromosome 21. And there are multiple phenotypic changes, and those changes can range from mild through to severe. Now, Down syndrome is interesting because it occurs later in the mother's life, in that if you have a child later in life, generally above 40 is the cutoff when people start getting interested in whether you will have Down syndrome. This is a graph showing the age of the mother, and this is the incidence of Down syndrome. And you can see under the age of 30 or even 35, it's very, very, very rare. 1 in 1,000 to 1 in 2,000, so it's quite rare. Once you get to above age 40, it's about 1 in 200, and after that it becomes quite significant. Say 1 in 50 and about your mid-40s up here. So why might this be? Well, if you think about where eggs are developed, and a female's eggs are held in prophase 1 since the time they were born. As a woman, you are born with all your eggs already in prophase 1 for your whole life. So by the time you release an egg at age 40, they've been sitting around for 4 decades. And one hypothesis could be that your chromosomes have been sitting in prophase 1, and the spindle fibers may not work. They may break. And if that's the case, then when anaphase occurs, maybe both sets of chromosomes go to one side. Now we have covered human sex determination. There are, of course, exceptions, as in everything. The female and male sex chromosomes, XX, XY, that's actually reversed in birds and butterflies, in that the males have 2 chromosomes the same, and females have 1 chromosome of each type. And they've been renamed as WW for the males and WZ for the females. And so the male is homozygous in that it has 2 chromosomes the same, and the female is hemizygous for both W and Z here. In moths, it is slightly different. In moths, if you have just 1 X chromosome, in other words, you are haploid, then you are a male. If you have 2 X chromosomes diploid, then you end up being female. And that's just for the sex chromosomes. If you are completely haploid and you're a B, if you have only 1 of every chromosome, then you're a male B. And if you have 2 of every chromosome diploid, then you are a female in Bs. And then even such changeable conditions such as temperature and environment will alter the sex of organisms such as fish, turtles and frogs. And of course this is much more variable here. We have looked at autosomes and we've looked at sex chromosomes, but there are other genes in a cell that are not in the nucleus. And mitochondria and chloroplasts have their own circular prokaryotic-like chromosomes. And so these chromosomes have genes on, and we have to take these into account. For example, mitochondria contain about 37 mitochondrial genes, and of course those genes are something to do with ATP production. And if those genes are mutated in some way, then not surprisingly, we have problems in energy conversion and energy production by those mitochondria. So if we look at some of the traits of mitochondrial mutations, we can see that there's developmental issues, short stature, loss of vision, so functional difficulties, motor skills are going down, seizures, uncontrollable muscle movements, and even paralysis of muscles here. So we can see these are fairly predictable issues when mitochondrial genes misfunction. Considering inheritance patterns for mitochondria, you only get your mitochondria from your mother. So here, imagine here are 2 cells. One is the egg cell, one is the sperm cell, and when these 2 combine, then the nuclear DNA from both is combined into the nucleus of the embryo. However, the sperm cell does not contain mitochondria inside the sperm head, but the egg cell does contain mitochondria in its cytoplasm, and the resulting embryo also has the same mitochondria because it is the same egg cell. And so your mitochondria came from your mother's egg cell. None of your mitochondria came from your father. And so in this particular diagram here, we have a trait that the mother has here. So this particular number 2 here has the trait. And she passes that trait on to all of her children. So this son, this daughter, this son, this daughter, all have that particular trait because her mitochondria are passed on to each of her children. Each of her female children then, each of her daughters, then pass on the same mitochondria to each of their children, in these cases over here. But because the male does not pass on mitochondria, no mitochondria, Whenever there is a male child, even though it has the trait, it does not pass that trait on to their children. So here and here. There are other ways to control gene expression. Genes do not work on their own. Most genes have influences from other genes or even by the environment to alter their expression levels. And so variations in gene expression can affect all kinds of pedigree analysis. So we're going to look at two terms that we use for affecting gene expression with the genotype and the environment. One term is called penetrance. And penetrance can be described as the probability that a disease phenotype, or any trait, will appear when that genotype is present. In other words, you have the genotype, you have the gene. Does that mean that you have the phenotype? If you always have the phenotype when you always have the gene, the penetrance is 1. If you say, if you have the genotype but only get the phenotype, say 60% of the time, then a penetrance is 0.6. So, and let's say if you have the gene and you only have the phenotype, say a quarter of the time, or 1 in 4 times, then the penetrance would be 0.25. And so a penetrance figure is the probability that you will have the phenotype if you have the genotype. An example for this would be breast cancer. If you have the double recessive BRCA1 genes, so you have the genotype for breast cancer, it has a penetrance of 0.8. In other words, you have an 80% chance of having breast cancer if you have those two double recessive BRCA1 genes. Expressivity is a variation on that, in that if you have a particular gene, how much of that gene is expressed with that particular phenotype. And so it has a range. You can have a low expression all the way through to a high expression of a particular gene. And so a gene may have low expressivity, or a gene may have high expressivity. And some trait, for instance, Down syndrome, for example, there are individuals with very, very mild Down syndrome, expressivity of very low levels for Down syndrome, and there are individuals with very high levels of expressivity for Down syndrome. You can combine both penetrance and expressivity in this diagram here. This top diagram shows only penetrance, and we see that out of 9 individuals, only 1, 2, 3, 4, 5, so the penetrance here is going to be 5/9. And here is this middle diagram is expressivity, and you can see the different shading, and here is very low expressivity, and here is very high expressivity, and you can see individuals have different levels of everything. And we can combine them so that in a population we have penetrance and expressivity, and here are some individuals with no expression whatsoever, and those that do express it have very high expression or very low expression, and here's some medium, here's some high, here's some medium, here's some medium. So we can combine both concepts. This concludes Chapter 4.